Great. Thank you very much. This is always a great meeting. I should say that I have some pathology training, but I'm an interventional cardiologist as well, so I do take care of patients. Let me see if I can. How do I advance this here? There we go. These are my disclosures. So the first point I want to make in this talk is that the SFA needs drug. This is a drug talk, and this is the results of the ISAR-STATH trial, which basically compared or randomized patients to paclitaxel-eluting balloon plus a bare metal stent, a balloon angioplasty plus a bare metal stent, or directional atherectomy alone. And you can see the results for freedom from TLR were superior for the paclitaxel-eluting technology versus technologies that do not elute any drug. Now, the types of drugs in humans above the knee, we know that sirolimus and its analogs have failed. The trials were unsuccessful. We know that paclitaxel is the main go-to drug. Whether it's delivered on a stent or a balloon, it's been successful. And these are the results of different trials showing you a negative trial, the everolimus-coated nitinol stent for SFA, the STRIDES trial, and the positive Zilver PTX trial. Now, why didn't everolimus or sirolimus and its analogs work above the knee? Well, we don't exactly know for sure, but we know that as the nitinol self-expanding stents expand over time, the interstrut distance becomes larger. The drug delivery becomes less effective. Sirolimus really needs the delivery from a platform. It does not have high tissue binding characteristics, and that makes it much less successful, especially if the interstrut distance is large, as in large arteries in the SFA. Paclitaxel has very high tissue penetration, especially when coated on balloons with excipient. They're very efficacious because paclitaxel crystallinity, greater penetration, and use of excipient allows for sustained release of paclitaxel. And paclitaxel devices are now the gold standard for the treatment of SFA. This is the DES trial showing paclitaxel-eluting stent versus the POBA plus bare metal stent, and the DCB trial, paclitaxel-eluting drug-eluting balloon versus POBA, showing both significant freedom from TLR with the paclitaxel technologies. However, there are no head-to-head randomized trials comparing DES to DCB for SFA disease, and there's a lot of questions clinically. When do I use a DCB? When do I use a DES? What is better? These are the drug-coated balloons that are currently in development. The red ones are the ones we have preclinical experience with. The green are what's approved in the United States. We have the Impact balloon and the Lutonix drug-eluting balloon. We conducted the first side-by-side preclinical study of the Impact versus Lutonix in the porcine model. We used 1x and 3x doses. We evaluated skeletal muscle and coronary band at 28 and 90 days in the porcine model. We did distal drug concentration and histology. I'm not going to go over all the details, but I'm going to show you the salient results. We evaluated distal emboli. Remember, of course, with drug-coated balloons, you want to make sure the drug gets to the target area and does not embolize to distal areas where it could cause damage. In the porcine model, you can basically evaluate these distal beds that are distal to the femoral artery, and I've listed them here, and you can look for signs of embolic disease. You can see here the downstream incidence of distal embolization in the study was significantly higher with the Impact balloon technology versus Lutonix, whether you use a single balloon or overlapping balloons both at 28 and 90 days. And more impressive was the paclitaxel tissue concentrations down below. You can see down here the skeletal muscle coronary band concentrations for Lutonix were significantly lower than that for Impact, whether you use a single dose or a 3x dose. The differences were even magnified further, showing you there is much more downstream paclitaxel embolized from the Impact technology. This is just showing you the histology. You can detect vascular changes, including basically dying nuclei embedded in homogeneous pink material. That's a yellow arrow. And you can see here there's fibrinoid necrosis in the black areas representing embolized paclitaxel. You can also see crystalline material lodged in the vessels themselves, consistent with embolized paclitaxel. Now, the significance of embolic disease is not exactly well known in terms of drug-coated balloon technologies, but there have been several case reports showing vasculitis after treatment with drug-coated balloons. I think the significance of this should not be undersold. Well, calcification, we tend to talk about peripheral artery disease and its treatment without calcification. It is nearly ubiquitous in the peripheral arteries, as you all know. The prevalence of calcification by CT angiography in 60 symptomatic patients was done on a clinical study. You can see they graded calcium by quadrant, whether it was 1 quadrant, 2 quadrants, 3 quadrants, or 4 quadrants. Most of the patients had 2 quadrants or greater calcium. So calcium and peripheral artery disease are run together. We did our own study, histologic study, in 10 asymptomatic legs, and you can see that we had, in terms of above the knee, we had 65% had intimal calcification in 1 quadrant, medial and 90% in 1 quadrant, and the rest had 2 quadrants or greater, but most of them had 1 or 2 quadrants. But this was in asymptomatic patients. Imagine what would be in symptomatic patients. And this slide was shown before. There's an inverse relationship between the primary patency and late lumen loss with calcium after 12 months of follow-up with drug-coated balloons. This is impact technology. You can see as the calcium grade went up in the artery, the efficacy went down. Late lumen loss went up, and the freedom from TLR went down, indicating problems with drug delivery, probably with calcium. Now let's move to stenting. Provisional stenting is actually required in a fair number of cases. It's underestimated. Even if you use a balloon, stents end up being used anywhere from 33 to 50% of cases. We did a study, a preclinical study again, comparing different technologies. A DCB alone, a DCB plus a bare metal stent, a POBA plus DES, or DCB plus DES. And we asked 2 questions, again, in the porcine model. How does a DCB effect compare with DES, as I referred to earlier? Is it safe to use DES after DCB? Those are the 2 essential questions of this study. We can look at markers of drug efficacy by pathology in these preclinical models by using these endpoints. Basically, you can tell if drug is deposited by looking at things like fibrin deposition, muscle cell loss, depth and circumference, and medial proteoglycan collagen replacement. Those are indicators that the drug has been there, done its job, and either left a repair area or is still killing some of the muscle cells, which is really the goal. And you can see in this study what we found was interesting was that the DES groups had much higher medial muscle cell loss, that is greater drug efficacy versus the groups that had DCB but no DES. So DES was superior in terms of drug delivery. It was also superior in terms of the circumference of drug delivery. You can see both DES groups had 4 quadrants or greater drug delivery, whereas DCBs only were able to deliver drug to essentially 25% of the area of the artery, and there were 3/4 of the artery undelivered, at least by the smooth muscle cell loss definition we used. Fibrin deposition was also the same. You can see the DES were far more effective at creating fibrin deposition than were the DCB technologies. Again, what we saw was findings of distal emboli with all DCB groups. You can see no matter which DCB group we look at, there was evidence of distal emboli in all those groups. The only group that did not have any evidence of distal emboli was the group that had only DES technology, no DCB technology, showing you that drug-eluting stents do not really embolize paclitaxel distally, but the DCB technologies do. So this was an example of histology just to show you the differences. And you can see here the DCB plus DES group. You can see that fibrin is basically circumferential in the entire artery. There is smooth muscle cell loss. There was good drug effect that was also seen in the POBA plus DES group. Same effects, amount of fibrin here. You can see in the DCB groups, either followed by bare metal stent or DCB alone, the fibrin was nowhere near as extensive as seen in the DES groups. The drug delivery was less, and the muscle cell loss was a lot less. There was a lot less with muscle cell loss, showing decreased efficacy. So in conclusion, in SFA lesions, self-expanding stents with sirolimus and analogs were ineffective above the knee, probably because of continuous expansion of the stent with time and decreased efficacy from poor drug penetration of sirolimus. Improvements in stent technology and use of paclitaxel, which is a cytotoxic drug with greater inhibition of proliferation and drug penetration, when applied on a stent or balloon, is efficacious, there's no question. Balloon coatings with excipient and paclitaxel are essential for the success of drug-coated balloons. Irrespective of preceding DCB or POBA, DES showed maximum biologic change in the neointermediate, suggestive of a superior drug effect with DES versus DCB. Distal emboli, as I said, were exclusively seen in DCB groups. DES plus DCB or POBA showed greater desired biological effect compared to BMS plus DCB or DCB alone, and we did not detect untoward effects from using a DCB before a DES. In severe calcified lesion with evidence of vessel dissection, prolapse, or angioepic unacceptable results following DCB usage, DES should be considered rather than bare metal stents. Thank you.